We're going to take a brief look at the Unit 2.2 Prokaryotes. It's worth noting that prokaryotes and many other things in biology can have their pictures taken by electron microscopes. The transmission electron microscope provides a relatively flat-looking picture, as you can see down here, whereas scanning electron microscopes provide these often beautiful and falsely colored three-dimensional images, as seen right here. Now, prokaryotes, in comparison to eukaryotes, are much smaller, about 1 to 3 micrometers in length, whereas eukaryotes can be anywhere from 10 to 100 micrometers on average. And they have some features about them that are worth noting. They break into two categories. Largely, the archaea and the bacteria are both considered prokaryotes. They're smaller and simpler than eukaryotes. They came around first in terms of evolution. They don't have any membrane-bound internal cell structures or organelles. They are always unicellular, and they divide by binary fission, which we'll go over in a moment. Now, the membrane organelles can be seen prominently in eukaryotes, but there are no membrane-bound surfaces in prokaryotes, which makes them a little bit easier to draw. I'll show that relatively quickly to you here. Start by drawing a pill shape, provide it with a few flagella, maybe 2 or 3. Give it some little hairs on the outside of the body. Those would be the pili, which I will not label here, but you can see on the next slide. Inside of that, I like to draw a little window into the inside of this bacteria. And just inside of that, provide a second line. The outside line would be the cell wall, and the inside line would be the cell membrane, which is on the inside. Inside, you can give a little swiggle to dictate the nucleus region, or I should say the nucleoid region. There's no nucleus, it's not got a membrane. And provide little dots for ribosomes, and of course the blank space all around would be the cytoplasm. You should be familiar with these, especially the fact that the ribosomes are 70S, and in addition, you should know their functions. The cytoplasm is a liquid that holds internal cell parts and enzymes for metabolism. The ribosomes synthesize proteins. The pili help attach to other surfaces and transfer DNA. The plasma membrane, or the cell membrane, controls what substances get in and out via pumps and a number of other processes. The cell wall is a protective coating that can be gram-positive or negative. The nucleoid is what holds the genetic material, and flagellum help it move. Binary fission is a relatively straightforward and simple process that involves copying the DNA, moving it to opposite sides of the cell, and splitting the cell into 2 clones that are identical. This happens very quickly and often results in exponential growth, as seen in many bacterial colonies. Done.